Now, in this second part of genetics, we will look at heredity, which is basically the study of how traits or characteristics are transferred from parents to offsprings during reproduction. In order to understand inheritance, we first need to understand the concept of homologous chromosomes. We said that in the body of somatic cells, chromosomes exist in pairs called homologous chromosomes. Homologous chromosomes are similar but not necessarily identical. Consider, for example, these pairs of homologous chromosomes in the cells of an insect. As you can see, they are of the same size and each has two genes on the same positions. The first pair of genes could, for instance, code for eye color, and both code specifically for blue eyes. So both are represented by letters small b. They are the same and identical. The second pair of genes could code for, let's say, color of wings. However, they don't code for the same color. The gene capital R could code for red wings, and the small r gene code for gray wings. So they both code for the same trait, color of wings, but different forms of colors. They are the same, but not identical. We say there are two forms of the same gene, and we call such pair of genes alleles. We also know that each member of the homologous chromosome is contributed by either of the parents during fertilization. In other words, sex cells or gametes have only one member of the homologous pair, and that is because the chromosomes segregate during metaphase 1 of meiosis. After fertilization, the homologous pair is restored. Now the question is, if these two alleles are present simultaneously in the body cells of the insect, what then will be the color of the insect wings? That is what we want to find out. An Australian monk, Gregor Johann Mendel, is credited for his pioneering work on the laws of inheritance. He carried out various breeding experiments with the garden peas and observed variations in different characteristics. One such experiment was crossing of a tall and a dwarf pea plant. Since the cross was between plants that differed in a single trait, that is height, Mendel called it a monohybrid cross. Further, Mendel called the tall and the dwarf plants of this experiment the parental or pea generation. After the cross between tall and dwarf plants, Mendel collected the seeds and planted them to produce the first hybrid called the first filial or F1 generation. He found that all plants in the F1 generation were always tall, like one of the parents, and the dwarfness had disappeared. Mendel conducted similar experiments with other pairs of traits and made certain observations. Every single time, he found that the F1 hybrid resembled one of the parents, while the other parent's trait was missing. To explore this further, Mendel self-pollinated the F1 hybrid to produce the second hybrid generation of plants known as the second filial or F2 generation. Interestingly, this time the progeny included both tall and dwarf plants in a definite ratio. 3/4 of the plants in the F2 generation were tall, while 1/4 were dwarf. Mendel also noted that the plants did not show any blending or mixing. In other words, no plants were in between the dwarf and tall plants. Once again, to confirm his observations, Mendel conducted experiments with other traits like the color of the pods. His results were confirmed when he found that both the parental traits were expressed in the F2 generation in the ratio 3:1. Again, there was no blending. From his experiments, Mendel concluded that something was being steadily passed from the parents to offsprings over successive generations. This was the reason why the deafness that had disappeared in the F1 hybrid had reappeared in the F2 hybrid. For lack of a better word, he called this thing factors. But today, we know that these factors are in fact genes. Genes are labeled with letters for our convenience. The gene coding for tallness is denoted by a capital letter, while the gene for dwarfness is denoted by a small letter. Because all plants obtained from a cross between tall and dwarf plants were all tall in the F1 generation, the tall trait is denoted by capital T and the dwarf by small t. Here, capital T and small t are alleles of each other. That is, they are different forms of the same gene. Both of them code for height of the plant. The two chromosomes are homologous. Therefore, the plant cells have one of the three possible combinations of the alleles. That is capital T, capital T small t, or small t, small t. Here, capital T, capital T small t, and small t are called genotypes of the plants, and the descriptive terms tall and dwarf are called phenotypes. The alleles in the genotype capital T and small t are similar and are called homozygous, while the alleles in the genotype capital T small t are dissimilar and are called heterozygous. Today, we know that genes are located on chromosomes and are transmitted from parents to offsprings via gametes. Body cells contain a pair of genes controlling each characteristic, but gametes contain only one of these genes. We can represent the inheritance of dwarfness and tallness diagrammatically using a genetic cross. This is the conventional symbol for male, and this one is for female. In the first stage, Mendel crossed a pure homozygous tall plant with a pure homozygous dwarf plant. Assuming that he took the pollen or male gametes from the tall plant and dusted it on the stigma of the dwarf plant. The parental phenotype, the male was tall and the female was dwarf. Parental genotype, the male is capital T, and the female is small t, small t. Then there is segregation to form gametes, and we have capital T, both of them circled, as well as small t, small t. Fusion occurs during fertilization, and we see capital T fuses with small t, and we get capital T, small t. And it's the same for all the others. So these are the genotypes of the F1 generation. As you can see, genotypically, all F1 generation plants are heterozygous. That is, they carry both genes for tallness and dwarfness in their cells. However, phenotypically, we saw in Mendel's experiments that all of them were tall. And therefore, only one character expressed itself in the F1 generation. This character is said to be dominant, and the one that does not express itself is said to be recessive. And so, the gene for tallness is dominant over the gene for dwarfness, which is said to be recessive. When the F1 plants were selfed, we had a dwarf plant. Let's see how that came about in a genetic cross. Parental phenotype, both male and female were tall. Parental genotype, both were capital T, small t. They segregate during gamete formation, and we circle the gametes. Fusion occurs, and we have the F2 generation. The first one, we have capital T, capital T, small t, capital T, small t, and small t, small t. Genotypically, we have one capital T, two capital T small t's, and one small t, small t. In his experiment, Mendel obtained 3 tall plants and 1 dwarf plant. And this is because in the homozygous capital T and heterozygous capital T small t, the gene for tallness, that is capital T, is dominant over the gene for dwarfness, and so the 3 plants will be tall. A recessive gene only expresses itself in the homozygous state, like in small t, small t. The dominant gene expresses itself in both homozygous and heterozygous states. If we crossed 2 tall plants, we shall obtain all tall plants in the F1 generation. Equally, if we crossed 2 dwarf plants, we shall obtain all dwarf plants. A Punnett square can also be used in predicting the outcome of a cross or breeding experiment. It is a graphical representation that helps to calculate the probability of all possible genotypes of the progeny in a genetic cross. For Mendel's experiments in the F1 generation, Mendel crossed a homozygous dominant, that is capital T parent, with a homozygous recessive, that is small t parent. The gametes produced by the 2 parents are written on the 2 sides of the square, and the capital T small t progeny produced by the fertilization of the gametes are written within the square. The monohybrid crosses are based on Mendel's first law, which states that the characteristics of an organism are determined by internal factors that occur in pairs. Only one of a pair of such factors can be represented in a single gamete. It is now known that these factors are actually genes, and that the process that produces gametes is meiosis. In conclusion, and from Mendel's experiment, we see that in pea plant, the character of tallness is dominant over the character of dwarfness in the F1 hybrid, and that both tallness and dwarfness appear in a ratio of 3:1 in the F2 hybrid. This is called complete dominance, and Mendel formulated the law of dominance, which states that in a monohybrid pure line cross between parents with contrasting traits, only one form of the trait, that is the dominant form, appears in the F1 generation, and both forms appear in a ratio of 3:1 in the F2 generation. Important to the law of complete dominance is the fact that recessive parental trait was expressed without any blending in the F2 hybrid. After verifying the results of the cross, it was found that the segregation of alleles is a random process, and the chances of a gamete containing either allele is 50%. Alleles do not show any blending, but instead segregate from each other during gamete formation into different gametes. And now, determining unknown genotypes. Now, because of complete dominance of the capital T gene in the garden pea plants, looking at the phenotypes cannot tell us the genotype of the plant. For example, you cannot predict whether a tall plant from the F1 or F2 generation is homozygous dominant or capital T, or is heterozygous dominant, that is capital T small t. A test cross, that is a cross between individuals of the F1 generation with a homozygous recessive parent, is carried out. If their offsprings are all tall, then it means the parent was homozygous tall or capital T. However, if it bears a mixture of tall and dwarf plants in a 50-50 ratio, then it was a heterozygous tall or capital T small t plant. And so in general, through his monohybrid cross experiments, Mendel proposed the laws of dominance and segregation. But does this law of dominance apply to all genes in all organisms? Hybridization experiments conducted on the pea plants were also conducted on the snapdragon plants. Apart from plants, experiments were also conducted on birds and on several animals like mice, rabbits, cats, guinea pigs, and even humans. It was then discovered that the law of dominance did not hold true in all cases. In other words, Mendel's laws were not universal in occurrence. Let's see the experiment conducted on the snapdragon plants. When a pure red-flowered snapdragon with genotype R was crossed with a pure white-flowered snapdragon with genotype r, the results were quite different. The flowers of the F1 hybrid were pink, and the genotypes were R. That is, the phenotype of the F1 plant did not resemble either of the parents but was in between the two. Then, the flowers of the F1 hybrid were self-pollinated, and the genotypic ratio obtained in the F2 hybrid was 1:2:1, the same results obtained by Mendel on the pea plants. However, the phenotypic ratio obtained in the snapdragon plant was also 1:2:1, which is different from the 3:1 ratio in the pea plant. Here, both genotypic and phenotypic ratios are the same. Therefore, when a dominant allele does not completely mask the phenotypic expression of the recessive allele in a heterozygote, a blending of both dominant and recessive traits takes place in the F1 and F2 heterozygotes. This is called incomplete dominance. And this is why the phenotypic ratios obtained in the experiment on snapdragon plants are different from those of the pea plants. It is the gene for flower color that controls the amount of pigment in the petals. Each allele is a code for a trait, that is, specific amount of pigment. When both alleles for pigment are present, the petals are dark red due to heavy pigment production. On the other hand, if none of the alleles for the pigment exist, the flower is white. However, when one of the alleles is present, only half the pigment is produced, creating a pink shade. The alleles are therefore exhibiting incomplete dominance, which is a form of intermediate inheritance where one allele is dominant but not completely over the other. Next, we look at codominance. This is a homozygous white bull with genotype capital W, capital W. And this is a homozygous red cow with genotype capital R, capital R. What would be the color of their calf in the F1 generation? Well, it is not white, red, nor a color in between. The calf's coat is a mixture of white and red hair. In this case, the F1 generation does not resemble either of the parent as in complete dominance, nor is it in between as in incomplete dominance. What the calf displayed was codominance. Codominance refers to a situation where a heterozygous organism has a phenotype that demonstrates traits of both alleles. Codominant traits are not blended, but are independently and equally expressed. And so in totality, there are three types of dominance: complete, incomplete, and codominance. And so the expression of any trait depends on whether the two genes coding for it exhibit complete, incomplete, or codominance. Consider the ABO blood grouping system. We said the plasma membranes of red blood cells have antigens on their surface. It is these antigens that determine the specificity of the major blood groups. The antigens may be A or B. It is the gene I that is responsible for determining the type of antigen produced by the red blood cells. This gene has three different alleles: capital IA, capital IB, and small I. Or for simplicity, we can call them A, B, and O. Of these alleles, A and B code for antigens A and B respectively, while allele O code for no antigen. Again, alleles A and B are codominant, and both are completely dominant over allele O, which is thus recessive. Now, does that mean a person has three alleles at the same time? No. Humans are diploid organisms and therefore possess any two of the alleles. Consequently, there are 6 possible combinations of alleles. Therefore, if the allele from one parent is A and the other is O, only A is expressed in the offspring because A is completely dominant over O. And so the red blood cell will have antigen A, and the genotype of the offspring will be AO, and the phenotype or blood group will be A. Equally, if the allele from one parent is B and the other is O, only B is expressed in the offspring, and the red blood cells have antigen B only, because B is completely dominant over O. In this case, the child's genotype is BO and the phenotype or blood group is B. However, if the allele from one parent is A and the other is B, both alleles are expressed due to codominance. In this case, the red blood cells will have both antigens A and B, and the child's genotype is AB and the phenotype is blood group AB. If the alleles are both type A, the offspring will be genotype AA and the blood group will be A. If both alleles are B, the offspring will have genotype BB and blood group B. If both alleles are O, the red blood cells will have no antigens. The offspring's genotype will be OO and the blood group will be O. Notice that in this explanation, we have considered the alleles in the gametes that fuse during fertilization. But remember, the parents could be homozygous or heterozygous for the alleles. The alleles are transmitted from parents to offsprings in the usual Mendelian fashion. For instance, a marriage between a heterozygous male with blood group A and a heterozygous female with blood group B will produce children of all the four blood groups. Apart from being a good example of complete and codominance, the ABO blood system is also a good example of a case of multiple alleles. Another example of complete dominance is seen in the inheritance of the rhesus factor. Rhesus factors are antigens found on the surface of the red blood cells. The presence of the antigen makes an individual rhesus positive, while its absence makes the person rhesus negative. The presence of the rhesus antigen is due to a dominant gene which is dominant over the gene for its absence. If one of the parents is rhesus positive, then all the children will be rhesus positive. If blood from a rhesus positive person is transmitted into a person without the rhesus factor, the recipient produces antibodies against the rhesus factors of the donor. This causes agglutination of red blood cells of the recipient. If a woman who is rhesus negative is married to a rhesus positive man, once she becomes pregnant, the child will be rhesus positive. Rhesus antigen can cross the placenta into the mother's bloodstream, prompting the mother's immune system to produce rhesus antibodies. When the antibodies get into the fetal circulation, an antigen-antibody reaction takes place and the red blood cells of the fetus are destroyed. This is called hemolytic disease of the newborn or erythroblastosis fetalis. Next, we'll look at sex determination. Now, the sex of an organism is determined by specific chromosomes called sex chromosomes. In humans and other mammals, the XY sex determination system is followed. Every cell in humans, either male or female, has 23 pairs of chromosomes. Of these, 22 pairs are homologous and are called autosomes. The remaining 2 chromosomes are sex chromosomes because they carry genes that determine the sex of the person. In females, the sex chromosomal pair will be XX, while in males it will be XY. The X chromosome is not the same size and shape as the Y chromosome. In males, Sperm cells are produced through meiosis with haploid or half the number of chromosomes. So in a sperm, the sex chromosome can either be X or Y. In females, the egg or ovum has only an X chromosome. When a sperm with X chromosome fertilizes an egg with X chromosome, the offspring will be a female with paired X chromosomes. If a sperm with Y chromosome fertilizes an egg with X chromosome, the offspring will be a male with X and Y chromosomes. The sex of a child is a matter of chance and depends on whether an X or Y sperm fertilizes the egg. As you can see from this genetic cross, the chances of a boy child equals to those of a girl child. And now we look at linkage. Now, an organism has a large number of characteristics controlled by many genes. Due to the limited number of chromosomes, each gene cannot be located on its own chromosome, and therefore, chromosomes must accommodate many different genes, each controlling a particular characteristic. Those genes found on the same chromosomes are called linked genes. All linked genes constitute a linkage group and are inherited together and therefore do not segregate during meiosis. All genes located on the sex chromosomes are said to be sex-linked and therefore are transmitted together with those that determine sex. In humans, there are very few genes located on the Y chromosomes which control characteristics that are exclusively male. These include premature baldness and tuft of hair in the ear pinna and in the nose. Characteristics controlled by genes located on the X chromosome include color blindness and hemophilia. These characteristics can arise in either males or females, but are more common in males. Red-green color blindness is the inability to distinguish red and green colors by some people. This trait is linked to the X chromosome. The gene that determines normal color vision is dominant over that for color blindness. That means female carriers of color blindness will have normal color vision. So, can we have a male carrier? No. Males only have one X chromosome, and the presence of the gene for color blindness makes them outright colorblind. If capital N represents the gene for normal color vision and small n the gene for color blindness, let us see the possible outcomes of a marriage between a carrier female and a normal male. Parental phenotypes: the male is normal and the female is normal but a carrier. Parental genotype: male we have X capital N Y, and female we have X capital N X small n. Our fusion between the gamete, we shall produce 4 possible outcomes. This is a normal female, a carrier female, a normal male, and a colorblind male. Hemophilia is another sex-linked trait where the blood of the individual takes an abnormally long time to clot. In the event of a cat, this results in prolonged bleeding and hence the term bleeder's disease. Again, it means a female carrier will be healthy and we cannot have a male carrier. If capital H represents the gene for normal blood clotting and small h the gene for hemophilia. Let us see the possible outcomes from a marriage between a hemophiliac man and a carrier female. Parental phenotype: the male is hemophiliac and the female is a normal carrier. Parental genotype: the male is Xhy and the female is XH X. Then we have the gametes, and when they fuse, we get a carrier female, a hemophiliac female, a normal male, and a hemophiliac male. Let's take an example. Jane is a healthy carrier of hemophilia and her husband John is healthy. Using a Punnett square, determine the probability of them having A hemophiliac daughter, B, a carrier child, and C, a hemophiliac child. And so Jane is a carrier, and that means she has the genotype XH X. And John is normal, and so he has the genotype XHY. Then we fill in the Punnett square. And now we label the phenotypes of the offsprings. This is a healthy daughter, healthy son, carrier daughter, and a hemophiliac son. And so question A, the answer is 0. There's no hemophiliac daughter. B, the answer is 25% or 1 out of 4. C, child with hemophilia, male or female. It's only 1 out of the 4, and so 25%. And now we look at family pedigree. Let us do an example to learn how to do pedigrees. Hemophilia is a sex-linked disorder. Mary does not have hemophilia, even though her mother did. Alan, Mary's husband, is hemophiliac. Their son Barry is healthy, but their daughter Rachel is hemophiliac. Draw a pedigree for this family. First, in pedigrees, females are represented by circles and males are represented by squares or rectangles. Shading means the person is unhealthy or has the condition. Half-shaded means a carrier. Obviously, for hemophilia, we do not expect a half-shaded square because there are no male carriers. We start with Mary. She doesn't have hemophilia, but she could be a carrier. We don't know. So we leave the circle unshaded. Her mother was hemophiliac, so a shaded circle. Mary is married to a man called Alan, who is hemophiliac, so a shaded square. They have 2 children. The son Barry is healthy, so we have an unshaded square. But their daughter Rachel is hemophiliac, and so a shaded circle. We can now find out the genotype of Mary. Let's start with her mother. She's a woman, so XX. In order to be hemophiliac, she has to be homozygous for the condition, and so she has to be X. Alan is a male, so XY. To be hemophiliac, he has to be Xhy. Let's leave Mary first. Barry is a healthy male, so XHY. Rachel is a hemophiliac female, so X. Barry got his XH from the mother and Y from the father. And so we can be sure that Mary had at least an X capital H. Rachel got one X small h, obviously from her father Alan, who is hemophiliac. It therefore means that she got the other X small h from her mother Mary. And so Mary's genotype is actually X capital H, X small h. She is a healthy carrier, and so a half-shaded circle. Other genetically inherited conditions include albinism and sickle cell anemia. The two are, however, not sex-linked and occur due to mutations. Albinism is a condition where the synthesis of the skin pigment, melanin, fails and is characterized by a light skin, white hair, and pink eyes. Such an individual is described as an albino. The genotype of an albino is homozygous recessive, that is small a. A carrier for the trait is heterozygous with the genotype capital A small a and has normal skin pigmentation. In a family, an albino child can only be born if both parents are carriers of the recessive genes or both are albinos. Therefore, the probability that any child born to carrier parents will be an albino is a quarter. The probability that any of the children is a carrier is a half. The skin of an albino person is so susceptible to sunburn, and the eyes are sensitive to bright light. Sickle cell anemia occurs due to gene mutation through substitution. Normal hemoglobin type A consists of two polypeptide chains. In the sickle cell condition, one amino acid, glutamate, is replaced by another, valine, in each of the two polypeptide chains of the hemoglobin molecule. The resulting hemoglobin, known as hemoglobin type S, is defective and has marked differences from the normal one as a result of this seemingly minor substitution. Inheritance of sickle cell condition is a case of incomplete dominance. If a man with sickle cell trait, that is a carrier, marries a normal woman, the probability that any of their children will carry the sickle cell trait is a half. Application of genetics. Now, throughout history, animal breeders have improved the quality of animals through selective breeding and only keeping offspring with the desired qualities. For example, dog breeders preferred dogs with different features. Some preferred for large size, some for small size, some for brains, and so on and so on.